Hi Shelley, and hi everyone
Hi.
to the Code Maven channel on YouTube. Um, my name is Gabor Sabo, and in this channel we are talking about all kind of programming-related stuff. This time, Shelley, who was one of my students at the Weizmann Institute of Science, is going to present her project, the background, and what she did, and we're going to discuss it. So hopefully we are. Have a better understanding of what this programming bootcamp for scientists can do for you. So, Shelley, please introduce yourself, and then we go from there.
Yes. So, hi, I'm Shelley. I'm a PhD student at Eran Segal's lab. So, this is a big lab, and Eran is mostly computational scientist. He's also a mathematician, and we work with big data. Most of the times. My project, I'm a biologist, so I came without programming background into a lab of big data. So it's obvious why I need programming as a cook needs his hands or something. But apparently how we worked so far is like I had another student that did the computational stuff and I did the biological stuff. And now this course really helped me to be a bit more independent and to take some calculation into my hands. So it feels really nice. It feels really good from my side. And it was a very good experience, the course for me. So I'll say a few words about our study because my project is really part of the study and I can't really explain what I did without a short explanation, short biological explanation.
Yeah.
um, okay, so now you see the PowerPoint screen, right?
Yes.
Okay, so we ask ourselves, we know that within our gut, we have this huge community of microorganisms. Actually, by now, the calculation is that you have as many bacteria as you have human cells in the body. So it's not neglectable. And they have a lot of importance to health and disease outcomes. So apparently as the human body lets all these bacteria sit within the body, and I'm talking specifically about the gut, but we have them on the skin and in other areas as well. So the immune system has somehow to coordinate with them. So it doesn't want any bacteria and not in any place. So it's very tightly moderated, and it is a very, very complex um, interaction between microbiome and the immune system. And we want to ask which antibodies the immune system is producing against the microbiome. And this is the majority of the project and what I will present now. So our system goes like this. We have all these microorganisms, the microbiome, and I chose a few proteins from them. So, for example, I picked here the brownish spike-like protein, and I picked the pinky one from here. And we took a company, and it synthesizes the DNA for us. And we actually take phages. The phages are just for us, they're just a scaffold to reproduce our proteins of interest. So we take the phage, each phage is making for me the protein I'm interested in here on the surface. Phages are types of viruses. Um, and then I take from humans blood, and the blood contains antibodies, and I mixed it up together. Some of the antibodies will recognize the proteins from the microbiome that are represented here. And with some other technique, I can actually take only the ones that are bound by antibodies, and I can know the sequence, meaning which protein they have on their surface. And then my output is actually who was bound. So out of the entire library, the entire repertoire of proteins, few of them will be bound by antibodies, and my output is now that I know which proteins from the microbiome are recognized by antibodies of the patients, of the people in the study. So we actually have this paper. We tried to publish it, and one of the reviewers said he doesn't trust the entire system, kind of, and he says, you know what, if this system is true, I want to see flagellines bound. What are flagellines? Flagellines are the, if you look at here at the purple bacteria here, it's like it's the proteins that allow the bacteria to swim, to move.
Okay.
So he said, okay, if you know to bind the bacterial proteins, to show the binding of bacterial proteins, I'm sure that you should see a signal for flagellas. So it is known that the human body produces the antibodies to bind the specific proteins that allow the bacteria to swim. Because if you think about it logically, it makes sense, because this is how you control the bacteria. You control the movement of the bacteria. So it's kind of a proof of concept. Show me you have some very reasonable result within your output. And so I took this as part of the final project of the course. And I will present now two data frames. By the way, I'm working with mostly with Pandas. Pandas is a very useful library for my type of data. So I'll present you the two data frames that I'm starting with, and the script, and then the output. So here you can see, now you see the first data frame of Excel, right?
Yeah, I see an Excel file.
So if you see here the numbers 0, 1, 2, 3, this is all the epitopes, all the proteins I have present on the phages. So if each row here will represent a phage that has one type of protein, and we have 244K ones, different variants within the library. And here you see the
44,000. Okay.
Yeah.
Okay. I wasn't sure that I was hearing the K there, and yeah. Okay.
Yeah.
And
Yeah, it's big data.
so you have it, all of them in this Excel file as well?
Let's go down. Yeah, with the header, it's 244 and 1.
Wow. Okay.
Sometimes our data frames are even so large you cannot open in Excel. So this is like the type that I can kind of even maybe sometimes work out without Python, but when it goes to more than 500,000 rows, it's impossible even to open. So you should know Python for sure, just for viewing even, or so the first data frame, we have here the number, just the number for the protein, the name of the protein, if we know it, we don't always know it, and the percent pass. This is already a result. So in how many of the people we checked, we actually got binding. We actually got the antibodies binding this protein, and we have the output. So this is the first.
I see like a 14 there.
14 is an example for something that is very reactive relatively. So 14% of the people.
Had a 14%. Okay.
Yeah, 14% of the people. We had 1,000 people.
Mm-hmm.
So 14% of the people recognize this protein.
Okay.
Okay, so this is the first data frame that I have. The second data frame should be, maybe it's this one. No. Okay, did it change now? This I'm sharing something.
No, you are not sharing yet.
Okay. So this should be the second one. The second one is our actually attempt to give a better identity to our proteins. So we went to a data frame that is called Uniref. It is kind of a well-known in biology. And here you see all the antigens. And through some blasting process that I will not talk about it because it will be too long. So we have now a good annotation for our proteins. For some of them, for example, for this one we don't. But it says that the number 5, it's myosin 7, for example.
Okay.
And I had, and actually the name is here until the first white space.
Okay.
So I had also to parse this. Okay, so at this point, I want to switch to the script. Basically, I'll say in advance what we want to do. We want to take the result, the percentage that it passed. We want to merge it to the identity of the proteins. I want to search for flagellas. And
So
I want to,
the word flagella, or what is
yeah,
that?
flag, because there is flagella and flagellin. So I went over this manually, and I figured out which term will catch most of the
Okay.
flagella's proteins. So, and I want to show the reviewer, you see, if I take the flagellas, and if I take our negative controls, which are just random proteins, that they're not real, they're not in the body, they're not coding for anything that makes sense. So if I'll show these two groups, you'll see that the flagellas are really reactive. Okay, so now I want to share the script. So I thought of, I don't know why I have too many things open. It doesn't, here it is. Okay, so let's see what I have here. Here I imported everything that all the libraries that I needed. Also for the graph, for the figure, I made such a graph, so I had to also have the library for that. We made some improvements, so I haven't imported time, and I have the time calculated, like how much time it is to run the script, but it's not that important. Okay, so let's start. Maybe we'll start from here. So we take the two input files and we open them, and then we concatenate the two data frames. And
Let's just look at the number of rows also, because to make it easier for someone watching this. So as I can see on line.
So maybe we'll start, I think, with step 1. I think it's okay. On line 45. So I read the two files that I presented to you, the input files.
Okay, which I see that you have the names in line 36 and 37. And then on line 35, you actually get to the path in which directory those files are, right?
So the way it worked now, it's actually how we wanted it, more for the course and maybe less for the real use. It's like that it works from the command line. So for this, the user has to provide a path, provided after he calls Python and then the script, and then he provides the path where the input files are.
Yeah.
And they should be under a specific name. And I have here warnings if it doesn't. So I mean, like if you don't enter all the requirements, so it will exit and write to you, save the 2 input files. It's what here on line 31.
You said that it was like you did it especially for the course. How would you do it if it wasn't for the course? I mean, it's primarily for you. So I was wondering what changes.
So it's here. It's actually this was my original line. It's just I had my own path. I just commented it out, and then I read from this, and then I ran it on PyCharm.
Okay, so what you say is that you would prefer for yourself to have a hard-coded path where your files are. After all, this is not something that you need to,
because it's not a generic. It's just one. It's one graph I want to do. Like if I have something that I every time go back to, so perhaps I would prefer also the comment line. But this is really kind of a one calculation, one time use.
What you might have do in this case is that to say that okay, take these 2 files and put the script in the same directory, and then you don't have to care about where it's located in the whole file system.
Okay.
And then you just assume that the script, the program, and the data files are in the same directory.
Okay.
Or somehow relative. So maybe your script is in the main directory, and there is a subdirectory called data. And if you want to make separate, if you would like to separate them, and then you put it there. And then still you say, okay, that has to be relative. This is in any case a very specific script program.
Yeah. Okay. Okay. So here on 41, I have another warning. So if you didn't, if the input files does not exist, so it also writes to you the same thing.
Okay,
and
I
then,
think that actually I think there's a bug here.
Yeah, why it ran? I tried it, but
but what I see is that the OR is inside the OS path exists. So it's a bug that it won't catch if some of the files if one of the files is missing.
Oh, okay.
I think that's the bug. So what you would probably need to write is that the OS path exists on one of the passes, and then closing the parentheses, and then or OS path exists on the other one.
Oh, like to all these phrases should repeat.
Yes.
Okay.
Because now you do the or first, and then I'm not even sure what the result of it is, true, false, or the probably, and then the OS path exists.
Okay, I kind of tried it. I tried to put just the first file, just the second file, none of them. So it made sense, but maybe I didn't catch it. Okay.
Yeah. Okay.
Okay, so then we go and open the 2 files that I showed you, and I concatenate them. They have the same index. This is why they can be concatenated
In Pandas.
in Pandas. So the name of the protein, it was like Agilent. It's just the number of the company, _1, 2, 3, 4. So this.
So as I understand that you add the columns together. So
yes.
So you'll have the index column is the same, remains the same, and then now you have more columns basically in this.
Yes. Okay. So unless they have the same index, it's getting hard to concatenate them correctly. So because they have the same index, I can do it easily. And then I had some and like some nuns or empty. I had to fill them up with 0 because biologically this is what it means, that we didn't get any binding there. So we refer it to 0.
Okay.
And then I went to parse the first column that I showed you from the Uniref. You remember that I said, okay, what's interesting for me, it's until the first white space.
Yes.
So if you go up here, you can see that it is called the parse column. So I go to the row of the Uniref function, and I split it until it has a white space n equals. This is how I get, and I take the first part. This gives me out of the whole line. Maybe just for a second I can.
Yeah, probably don't need actually the n equal there, or did you need it? Maybe
Yeah, yes, because sometimes I had a white space between the words and I wanted them.
Okay. So the name had a white space in it, yes.
Yes, so exactly. So like so I needed this character, this white space n equals. This is where the name of the protein ended. So this is what I chose to parse it. Okay, so we are here, and then I had.
I mean, you can show the Excel if that's what you wanted earlier. Do you want to do that, or?
I can do it. Just a minute. Okay, so for example, if you see at line 3, I have few white spaces that I don't want to cut. I don't want the possible. Yeah. I want the possible conserved membrane protein, and then this n equals something is the end of my name. Okay. So this is why so this is what it actually does. It takes the whole row and cuts it until the white space before the n equals. Okay, so let's go back here. Okay, and then I select the flagella antigens. So we'll go up and see how we do this. This is defined here. So antigen type. So I have now few rows that are relevant. If I have the word random in the column named full name, it means that it is my negative control. If I have flagella in the parsed function that we just created, I return flagella as antigen type. And if I also have a flagell in the full name of the protein, I also consider it a flagella. And else, I said return an empty string. Okay. And I will show the data frame that it is creating at the end, and it will be clear. Afterwards, what I want to do, I want to create my data frame that that contains only the antigen type that is not empty string. So out of the whole library that we have 244K, I'm just taking something that is my sub library, that is my negative control, and the flagellas. Okay, and I discard the rest. I think this is a good point to show the data frame that was created. And then I think this is it. No, let's see. What you see now,
there's the Excel file that the second Excel file or which one? One of the Excel files you showed.
Okay.
Percentage.
Okay, so now you see also the type of antigen here.
Yeah, so now this is the combined one that you also save to an Excel, right?
Yeah, so it has something. So first, we don't have the 44K rows anymore. it is just the ones that were identified either like flagella or negative controls.
How many
Okay,
are?
so we have here, let's see, 2066.
Okay.
Minus the header. So
Yeah, okay.
So here you see we don't have all the rows. We have only the ones that we looked. We have the full name. We have the percentage passed. Now we have zeros instead of empty things. We also did that. We have the Uniref function originally, the parsing that takes only until the n equals something. You can see it here, and the type of antigen. So it's either flagella or a negative control. Now it will go back to flagella. Okay, so this is actually the script result, and now I want to plot it. I want to give some description as to how these groups are different from each other. So I did it in two ways, actually. One is just a graph, it's a plot. And I think this is how it starts. A box plot. So I chose here in 66, you can see, I chose the label for the Y, the label for the X, the title for the graph, and so on. And the other thing which was nice, that is also an output of the script, it's kind of a descriptive statistics of this type of antigen column. And I saved these two output files. I saved them. This is here. This is the saving in 73. This is the saving of the figure, and in 77, this is the saving of the CSV.
Yeah, that we just see, right?
No, it's the statistical.
Oh, the statistics. Okay.
Yeah, statistics. And once I was like trying to improve a bit the performance of the script, so I wrote at the end some command of print the project is done and the time that it took. So
Yeah.
this is how the script ends. And now we'll see the two outputs. Okay. So this is the graph we get. We see the negative control, we see the flagella, we see the header, the Y, the X. So you see that in the flagella group, I have much more, each circle is one of the reactive antigens, and in how many people it reacted. So the negative control, you see it ends around like 1%. This is like the most reactive negative controls we have. And you see that in the flagella group, we have much more reactivity. So this is one way of showing it. The other way of showing it is the statistics. So you see here the percentage passed and the type of flagella and negative controls. And you see here like the numbers that, for example, the maximum of flagella, the most reactive one, was 13%, and the highest was here, half of percent. So this is kind of, and you have here the count, the mean, the standard deviation, the minimum. So it is also kind of a descriptive way to show how these groups are different from each other. So yeah, so this is basically it. I hope I kind of told it in a way that people can understand.
Yeah, I think so.
And yeah, I just want to say once again that for me it was like super important and really useful. This is just an example, but I have many scripts from this kind that I need to merge data frames, to sort them, to create new columns based on the data in another column. This is like really, really important for me, and I can be more independent now. If I have an idea, I don't need someone else to check it for me.
Yeah.
Even if I do it in like a clumsy way and I don't write the best way, but I'm independent and it's good feeling with my own data.
Okay. Great. So thank you for the explanation. And thank you everyone for watching the video.
Yeah.
Hope I don't know, enjoy your rest of your PhD.
Thank you. Okay.
Okay.
Bye-bye.
Bye bye.